Good evening, good morning, good afternoon, whatever it is, whatever time you're watching this. It's Ken's Talk meeting his deadline, episode 5 of the Human Genome Sprapp. 2-3 days in a row we are now doing videos. That's good, trying to do this every day. Got some cool looking graphs being generated off this data. I'll tell you what got me interested today. I did a couple things I learned today. First was I took a look at Power Pivot and started to import a database into Power Pivot, learning how that works. That's going to be the end-all, be-all solution to all this, is getting everything in Power Pivot. Until then, I'm still working with some data that the guru of Excel, Mr. Excel himself, Senor Excel, gave to me. He sent me a file with a bunch of genome data in it, and you know, it looked pretty cool. I don't know what it was from, and as I started analyzing it yesterday in episode 4, I realized that there's a 420 nucleotide or nucleobase string that is repeating through this 100,000 nucleobase set of data. It's 420 characters over and over and over and over again. That's what I found out yesterday in the middle of this video. Well, turns out that I decided to do a little bit more information digging into that, and here's what happened. Let's see if I can go through as fast as I can, because I don't want to spend too much time on this, but we'll show you what we learned today. So these are some really cool graphs that are showing information about this 420 character gene. I'm going to show you how I'm able to, I'm assuming it's a gene, but it's just a character of nucleotides. So all you do, a cool little formula you can do is to get all of these bases into one cell to be able to look it up on the BLAST database that the National Institute of Health has, which is here, which is, you can start looking for humans or mice or rats or proteins or all kinds of different things that you can search nucleotide sequences for. What I'm going to do is I'm going to go back into here and I'm going to show you a quick little formula. So I just want to make basically all of these show up in one cell in an order so that I can paste it into actually, I wonder what happens if I pasted this. Let's try that real quick. I haven't even, nah, I'm not going to do that. I'll show you the other way first because it's kind of nice. Alright, so the first thing you do, first cell, this equals this, that's fine. Then you say equals this and percent sign this. Then you have the same idea all the way down. You just pull this formula down. You're done. All of a sudden it's going to pick up the next letter and add it to it. So we need to take this sucker all the way down to sequence number 420, because I've already mapped this out yesterday, so I know where that's exactly where it ends. And voila. And what you get is a bunch of characters down here. This is this sequence for this repeating 420 character sequence. I'm going to copy it. I'm going to go to BLAST, and I've already done this, so I'm not going to be surprised. That's our string we're looking for. Okay, perfect. Let's go ahead and blast away. It's going to run, and it's going to say, guess what? Nothing exists, loser. No significant similarity found. For reasons why, I looked at the reasons, they don't make any sense to me. What did you send me, Mr. Excel? I want to know. I was trying to use this to figure out what area of the, I assume it's the human genome, what area of the genome this came from. Either we found a new gene, this is part of the junk DNA, or Mr. Excel is sending me rat genome and not telling me. I don't know. He's not a dirty rat. He's a good guy, so I don't know what he's doing to me. We're going to have to find out. I can't find it on BLAST. Can't find it here through the 7,000 genomes or whatever that they have in this database. No dice. So, well, I started to figure, well, let's start digging in and see exactly what this is. So this 420 character set, it just repeats over and over again through the 100,000 nucleotides. I decided to take a nice slice of this pivot table out, and I decided to grab just this 420 character gene, and that is what we're looking at here. I got two tables. One is density, and the other is mass. Those are the only things I'm looking at. These are just the mass of the nucleotides. If you open sequence 1 is a G. G has a mass of 151, moles to the grams per mole, something like that, right? Grams per mole, yeah, that's what the actual measurement is, right? Go back here. At grams per mole, and the density grams per centimeter squared. So this is the density here. It's the same idea here. And these graphs are going to show you higher areas of higher density, this one, and higher area is of higher mass. And I've got a few different visuals in this, and Excel provides many different options for visualization that are fantastic. This one here is just a normal line graph. So where it dips is going to be areas of less mass. Less mass. What does less mass mean? I don't know. What does it mean when it's more massive? These areas, these kind of holes at the bottom here are showing areas where it's more massive. Nucleotides more massive. This is kind of like a barcode graph. Same idea. Peaks and valleys. The valleys would be weaker, essentially, same thing on the density area. These valleys are going to be less dense areas. One of the graphs I really like is the radar. The radar graph ends up taking a it's a circle of all the 420 characters, and it starts in here and goes around in a clock, and then ends here. And it's the same idea as the bar graph or line graph. Areas that are higher density on this one and higher mass on the other one are going to be out towards the end. So we see here, oh, well, this is an area, this area right here in the middle, it's less dense. This is the way that the protein folds, or if this could somehow be modeled into the way that this protein, if it does make a protein, is modeled and copied. Perhaps a lot of these radar type looking things are all wrapped in together, and this area here would be a weaker area of the protein because it's not as massive and not as dense. Just speculating, but these visuals help you kind of see what is this 420 character gene, how does it map itself out, what are things to look for, I don't know. And why doesn't it exist in the database? That's something I'd like to know. So I've been able to pump these bits of data in here. Very cool. This is very, very cool. And you could do this with any gene, but the thing is repeating, and I want to know what it is. I want to know what it is. I want to know what chromosome is. I want to know if it's human or something else. Somebody get a hold of Bill John and find out where he got this data. I hope I didn't send it to him. If I did send it to him, then it's chromosome 1 for the 2003 data. But I don't think it is because I've seen the ends in here. All I see, how do I do a nice little check to ensure that there are no blank nucleotides and they're all A, C's, T's, and G's. I can just go up here and add a slicer for the base. When I add a slicer for the base, if I see anything other than A, C, T, and G, which I do not, they're all A, C, T, and G, then I know that there are no ends, and that's not the data I sent in. This is other data, so it's coming from somewhere else, and I am intrigued, Mr. Excel. I want to know where it came from, because we can't find this gene. Maybe we just found a new gene. That would be awesome in episode 5. Super, finding new genes. Way to go, because a sequence of 420 characters is pretty darn big. I know that these genes are thousands and millions, but it's still big. It's not 50, it's not 10. It does seem to really create a pattern in all this data. So you'd think that it would have been found already, but I would like to know what it is. This bioinformatics industry is a brand new industry, and you're going to be discovering things all the time. Plus, there's this variation in everyone's genome. I was thinking about this when I was looking at chromosomes yesterday. All the DNA testing that has been locking people away for the rest of their lives, I think it's generally done with the banding technique, comparing the DNA sample that's run on someone and the bands that are created on the chromosomes. They don't do a one-to-one molecular comparison, which is what this is. These are molecules here. And this thing is billions of characters long. They do not do this for every single person who murders somebody in the back of a Wendy's. They don't do that. I think they do the band testing, and the band testing leaves a lot of, a lot more variable error, where somebody's bands could essentially line up, but because you're not digging down into the specific molecules and the nucleotides that are contributing to those bands, then you don't know if it's exactly the same person's DNA. It seems like that would be a huge issue. I don't know. Things we're going to find the answers to as we keep going day to day. So, very cool visualizations. Love the radar graph. I'm going to start doing this with different sequences of genes that actually been sequenced. And I'm working with Power Pivot. That was my other goal today. Got into Power Pivot a little bit. Figured out how to put stuff in these databases. Have figured out how to write sequencing code in here. That is going to be a serious hurdle. I'm going to have to figure that out. But this active, with the table you're seeing now of visualizations can easily be done in Power Pivot. Of course, it's going to be a lot more jumbled. So you have billions of records, so when you break it out by sequence, it's not going to look like much. And you have to find this type of self-similarity, these type of patterns, this type of repetition, because if I were to look at the full 100,000 nucleobases, I would see this pattern repeating, 100,000 divided by 420 times, right? That's what I'm going to find. 2 and 2, 3 divided by 20, and you end up with seeing this pattern repeat. I think I didn't type in enough zeros there. Pretty sure I typed in only 10,000, yeah. So you're going to see this pattern repeat 238 times, and that's going to be a lot harder to identify. So there are going to have to be tricks we're going to have to write to make it do this, and then combining all these things together. So very cool. I like the pictures. I like where we're going, and hopefully we found a new gene or not. Hopefully I know where that gene comes from. Bill Joe, please let me know, man. I'll send you a YouTube comment and figure this out, because I am intrigued. So that is the Genome Sprapp here. How many C's are in here? How many A's are in here? Oh, the power pivot. What A's and C's? What about that, right? Interesting. What about A's and T's? Because those guys are the ones that bond. Yep. You can see how quick you just pivot. That's the beauty of the pivot table. Just pivot right there on one shot. So that's HGS episode 5, episode 6 coming up tomorrow. We're going to keep this rolling every day. Keep learning something every day. Eventually we're going to figure out the secret to human life, right? That's the plan. We'll see. Alright. Ken's Dog out.